Okay. All right. So last time we did operons, which is regulating transcription in prokaryotes. We specifically talked about the different combinations of operons that you can have, or regulated operons that you can have. Negative inducible, positive inducible, we went over in detail, and that's because the example of a negative inducible operon is the Lac Operon. And if you were asked, you know, how is the Lac Operon regulated, the answer is technically negative inducible. Positive inducible does play a part, but that is a secondary thing when we're talking about the Lac Operon, and that's because this is a secondary thing regulated via the catabolite activating protein, the capped protein. So, you know, if you want information on the capped protein, it's right here. This is positive inducible control. Usually this happens after the repressor on the Lac Operon has been removed. You have a mechanism to attach an activator and inducer protein to speed up the transcription of the Lac Operon. Not going to go over it in detail, but we talked about how the Lac Operon is potentially one of the potential extra credit questions you could have, and you know, you have to talk about, you know, what is the purpose of the Lac Operon, how is it regulated, both the negative part and the positive part, and what happens, you know, if there's lots of glucose or not a lot of glucose, what happens if there's lots of lactose or not a lot of lactose. And, you know, just like in the previous extra credit question, just give me enough information to show me that you know what you're talking about, and I usually give you full credit. I usually only take points off if there's just blatantly, like, so far wrong type comments that I can't just let it go. Let's see. All right. You don't need another tryptophan operon for this class, although the information is provided for you. You don't need to know for the exam, is what I should say. And now we get into the second part of the transcription information, and that is transcription in eukaryotes. So we learned about transcription in prokaryotes. We learned, you know, if you want to compare and contrast this stuff, which I highly suggest that you do, transcription in prokaryotes is executed by one enzyme called what? RNA polymerase, that's it. Just RNA polymerase, no Roman numerals, no Greek letters or anything. Although RNA polymerase in prokaryotes is made of several subunits, so you have to know about the subunits of RNA polymerase in prokaryotes, which are alpha, beta, beta prime, and sigma. You need to know those subunits are involved in prokaryotic RNA polymerase. All right, once you study your prokaryote stuff, now you can contrast that with eukaryote stuff. And so transcription in eukaryotes is actually carried out by 3 different polymerases. RNA polymerase Roman numerals 1, 2, and 3. And you may notice that this is pretty confusing, right? Because, you know, a lot of people will very easily get this mixed up with DNA polymerases in prokaryotes, right? What do they call those DNA polymerases in prokaryotes? DNA polymerase Roman numeral 3 and DNA polymerase Roman numeral 1. So here's another point in the notes where I say make sure you separate your notes into different pages and you can compare them to each other. Yes, question?
I'm sorry, did you say that the eukaryotic transcription, wasn't it like the alpha, beta, beta prime?
That one is prokaryotic transcription, is via RNA polymerase, no extra name attached to it. However, that one RNA polymerase has subunits with Greek letters. So yeah, that's prokaryotic transcription. So yeah, again, in the format of a lecture, it's very hard to show you all these differences because we can only go through things one at a time. That's why when you're going through your notes, separate everything in the different pages so you can compare them to each other. So on this section of the notes, in transcription in eukaryotes, transcription is carried out by 3 different RNA polymerases called RNA polymerase Roman numeral 1, Roman numeral 2, and Roman numeral 3. The color coordination here tells you which one is responsible for what transcription, meaning RNA polymerase Roman numeral 1 is responsible for transcribing rRNA, 2 is responsible for mRNA, and 3 is responsible for tRNA. Okay, so you do need to know that, and that is eukaryotes. All right. Common mistake or a common exam question is, which of the following polymerases is responsible for transcribing mRNA in prokaryotes? Half of the people will say RNA polymerase 2, but the question is saying which enzyme is responsible for transcribing mRNA in prokaryotes. The answer would be what? Just RNA polymerase in prokaryotes. Yes?
There's a graphic of the prokaryotic polymerase, and it has the CT details, but it doesn't say anything about that. Do you need to know anything about that?
The C-terminal domain on there. Let me see.
He said something about their function, but I think he was talking about eukaryotes.
It is more important in eukaryotes, so let me check, just make sure he doesn't mention it in the prokaryote stuff.
Yeah, that picture. Right?
Yeah, where was it? I just had it. Yeah, the CT domains.
Yeah.
Let me make sure. Yeah, that is more with eukaryotes. Which, did he already get all the way to that? That's quick. Okay. Yeah, that is more of regulating eukaryotic transcription, which we'll get to. I don't know, is it here or later? But we'll get to it when we talk about it. But yeah, the CT domain is a special kind of like control mechanism for transcription in eukaryotes. But for prokaryotes, I was never taught anything about that specifically, and I've never seen it pop up.
But they are present.
Probably. I don't know enough detail about that to know if it's similar in terms of regulating activity as it is in eukaryotes. And all I can tell you is I've never seen it asked about on the exam in the context of prokaryotes.
Okay.
All right. Other facts about RNA polymerase are listed here. It is also a multimeric protein. It also has 2 large subunits. Very similar to beta and beta prime, but notice they don't give you the name of these subunits, so therefore it doesn't matter. You may notice this pattern everywhere in this class where prokaryotes, they give you tons of information. Eukaryotes, they only give you a little bit. That's because you only need to know a little bit about eukaryotic stuff. Question?
You said, it says right there all have 2 large subunits that are similar,
Mm-hmm.
but then it's only in red, similar to beta and beta prime, so is that the only RNA polymerase 1?
Oh, I don't think that's on purpose. No, I don't think the red down here is the same thing as the red up there. Yeah, no. They don't even tell you which polymerase they're talking about here. That's, again, the fault of eukaryote stuff not being explained in detail in this class, and I personally don't know enough detail to know if every single one of these has a beta and a beta prime or not, and I've never seen it asked in that much detail before, so yeah.
So all 3 have the 2 subunits?
Probably. That's what it says here. Again, these are way more specific than anything I've ever seen given on the exam. If, in the rare instance, it is that specific on the exam, I will go over it during the exam review. But for now, just flat out what it says on here. It sounds like 2 subunits are what you see in all 3 forms of this polymerase. That's a really specific detail that I've never really had to memorize myself over the years, so I can't give you more information than what is stated up here. Like, I will be blunt with you with what I know you need to know in detail and what you just flat out have to memorize. This is a memorization thing. Okay. Let's see. Oh, did he go over this story here about this inhibitor? So there's a particular component in some mushrooms. I guess it's called alpha mannington. Basically, this thing is really specific for RNA polymerase 2. Leads to very painful death by poisoning because you are literally inhibiting transcription with this thing. All right. Let's see. Okay, before we get to this, let me go to this. Is it this picture? This picture. Okay. If you understand this picture, and I will break it down, it's basically all you need to know about the mechanism of eukaryotic transcription. It's just this picture. There are little components that are given in detail in 29b, but overall, this is what the mechanism looks like. So, here's how eukaryotic transcription works. And this is a lot of what my research focuses on. So, I will try to simplify it. All right. Here's a gene over here in green that needs to be transcribed. In prokaryotes, we learned that there was a set of sequences called promoter sequences or consensus sequences in prokaryotes, where those consensus sequences in a promoter region, their main job, their main function is to provide like a landing site for RNA polymerase to bind to and know where to begin transcribing. In eukaryotes, we have a very similar mechanism. We have a region just upstream of this gene called a promoter region, and you'll notice they have two different names here: core promoter and regulatory promoter. I'll go over that in detail in a little bit, but they both serve basically the same type of function that you see in prokaryotes. It's a region directly upstream from this gene, meaning these will not be transcribed. I'll go over some exceptions in a little bit. These will not usually be transcribed, and they contain sequences, base patterns of which transcription factors will like to bind to. So there's a term here that is used only for eukaryotic transcription. That is the phrase transcription factors. You see everywhere they see TF, TF, TF. Each one of those, and there's lots of them here and a bunch that are not depicted here, these proteins are necessary for activating and regulating transcription in eukaryotes. So there's a few listed here: transcription factor 2D, transcription factor 2B and A and all that. Don't worry about memorizing any of these. Probably in a genetics course you have to know which one does which. In this class you just have to know that collectively these are special proteins necessary for activating and regulating transcription in eukaryotes. How do they do this? Well, on the individual transcription factor scale, they each have a particular consensus sequence they like to bind to. They each have a different one that they like to bind to. So TF2D is a transcription factor that likes to bind to a consensus sequence called the TATA box. Remember back in prokaryotes, there was a consensus sequence that also had a name. I think it was called like Pribnow box or something back in prokaryotes that had a particular pattern for RNA polymerase in prokaryotes to bind to. In eukaryotes, the TATA box, so those are two different things. Pribnow box and TATA box are two different things. The TATA box is a consensus sequence in eukaryotes that transcription factor 2D likes to bind to, and if it finds an area in the genome with the TATA box, it will bind there. Just this transcription factor is usually not enough. You need all these other transcription factors, at least one more, to come in and bind to its respective consensus sequence that we don't need to know the pattern of. Basically, the way transcription works in eukaryotes is when a group of transcription factors bind to their respective consensus sequences in a promoter region, they recruit the signal for RNA polymerase 2 to come in. So contrast that with prokaryotes, where RNA polymerase 2 itself is looking for consensus sequences to bind to so it knows where to transcribe. In eukaryotes, the thing doing the looking is the transcription factors. The transcription factors are looking for consensus sequences to bind to. RNA polymerase 2 will just go wherever the transcription factors are going. Okay, RNA polymerase 2, and this is true also for RNA polymerase 1 and RNA polymerase 3, which are not depicted here, but they will go where the transcription factors go. Then you have this whole mess here where you have transcription factors, RNA polymerase 2, and notice there's different sizes of transcription factors, indicating a variety of functions and consensus sequences. But when they're all together, you form what's called the, where is it here? Basal transcription apparatus. That's when everything is put together. It is the transcriptional complex that is now ready to go and transcribe everything. Okay? So, the basics of this again are, there are consensus sequences here. There are transcription factors that like to bind to them. Once they're there, RNA polymerase 2 will associate with these transcription factors and then begin transcribing the gene. Eukaryotes are slightly more complicated than that. That's just the basics. Here's some extra complications. I'm going to zoom out a bit. All right, this yellow part is a region directly upstream of the gene called the core promoter. This is usually a distance anywhere between one base pair upstream of the gene to, I think, about 1,000 base pairs upstream of the gene. So this is about 1,000 bases wide. And any transcription factors that like to bind here are core transcription factors. However, eukaryotes regulating transcription can occur at sequences that are really, really far away. Moderately far away, we have a region called a regulatory promoter. There are consensus sequences there as well, and transcription factors will bind there as well. But they're quite far away. They're more than 1,000 base pairs away from the transcription start site. You do not see this in prokaryotes. In prokaryotes, regulatory sequences are usually very, very close to the transcription start site. But in eukaryotes, we have some that are far away. How do they interact? How could they possibly interact with the transcription start site? Well, take a look at this picture down here. Here's a theoretical transcription factor that is instead binding to the regulatory promoter region. It's pretty far away. Notice that it can interact with everything else through an intermediate called a co-activator. So, in prokaryotes, you learned about terms like co-inducer and co-repressor. In eukaryotes, annoyingly, they use very similar terminology, but it's a different thing. There are molecules called transcriptional co-activators. Their job is to make it easier for transcription factors to associate with the basal transcription apparatus. So notice how far away this transcription factor is. It is still able to interact with this huge thing here via an intermediate co-activator. Okay? The co-activator itself doesn't bind to any sequences. Notice that it's not touching the DNA. However, it helps a transcription factor that is far away to associate with the basal transcription apparatus. So that's two places that transcription factors can bind to in eukaryotes so far. One is the core promoter, and the other one is a regulatory promoter that's slightly further away. The last one, and this is where tons of research is being directed to because we don't know much about it, are these regions called enhancer sequences. These are so far away and have such a variety of patterns and distances away that we don't really know exactly how they affect transcription. We know the basics. We know the basics that there are sequences that can sometimes be 1,000,000 base pairs away that somehow still affect the transcription of this gene here, even though it's 1,000,000 base pairs away. How does it do that? Well, step one is as a transcription factor will bind to this enhancer sequence. How? There's a consensus sequence that just happens to be here. And as long as it's there, a transcription factor that matches with it will bind to it. Eukaryotic DNA has this feature that is illustrated here, in that it can wrap around like this. So even though this enhancer sequence is 1,000,000 base pairs away, because eukaryotic DNA can wrap around like this, notice how close this transcription factor can actually be to the basal transcription apparatus. That is a feature unique to eukaryotes. Prokaryotes cannot do this. There are no enhancer sequences in prokaryotes. So more comparing and contrasting for you guys. Bottom line is, think of enhancers as kind of like promoter sequences, just really, really far away. And there's even more special stuff with enhancers, which we'll go over in 29B. But these are the basic components that regulate transcription in eukaryotes, all right? So, big summary again. Here's a gene over here. In order to transcribe it, you need transcription factors to bind to their consensus sequences. Most of the important consensus sequences are in the core promoter. However, some can be in the regulatory promoter, which is slightly further away. And some can be in enhancer sequences, which are super, super far away. But once everything comes together, you can transcribe a particular gene. Now, eukaryotes are pretty complicated. It turns out that you could have many different transcription factors regulating this gene at different times, at different cellular conditions. Maybe, for example, if in the cell it's existing in it's exposed to a low oxygen environment. I'm kind of being biased because that's my research. It's in a low oxygen environment, and when it's in a low oxygen environment, maybe there's a transcription factor called HIF1 alpha that is only active in a low oxygen environment. That transcription factor will bind to whatever promoter regions that has its consensus sequence and transcribe those genes. Therefore, those genes will only be active in a low oxygen environment. Eukaryotes have this crazy thing where certain genes are active based on certain conditions, certain stimuli that are received by the cell. And it is regulated, the expression of genes are regulated simply by the enormous variety of transcription factors that you have that will bind or not bind depending on what conditions the cell is in. So, bottom line, look at this diagram if you want to learn eukaryotic transcription. This is probably at least 50% of what you need to know about eukaryotic transcription is on this one diagram here. Okay? Yes?
Do those all remain bound as transcription proceeds and
Oh.
finishes?
Good question. No, they similar to prokaryote transcription, they're just there at the beginning, and as soon as RNA polymerase is there, it's going to go and transcribe everything on its own. So they're there to set everything up, just like in prokaryotes you needed the sigma subunit to set everything up. Other questions? We'll go over the details in 29B, but this is an overview. All right, so let's see. All right. Some basic biology stuff.
Mitochondria is powerhouse of the cell.
Yes, that's right. The mitochondria is the powerhouse of the cell. Too bad that won't be a question in this class. But yeah, simple cell diagram. This is brought up because eukaryotes are special, right? There's a nucleus where all the DNA is. Transcription has to occur in the nucleus. And, you know, I guess I should be careful about this because both of these will appear as answer choices. Technically, you have to know that our RNA is specifically only made in the nucleolus. So it's stated here, but I don't want to accidentally give you too general information sometimes. But our RNA is specifically made in the nucleolus. Everything else can be made just in the nucleus, mRNA or tRNA. We know that because transcription occurs in the nucleus, but translation occurs in the ribosomes, which are out in the cytoplasm, that there must be a procedure for mRNA to leave the nucleus. And it does exit through nuclear pores, and there's other researchers elsewhere that figure out what regulates mRNA's ability to leave the nuclei. Who cares about that in an undergraduate class, so I'll move along. Let's see, what else do we have here? Nope. Yeah, remember this? This was that drug that, specifically inhibited RNA polymerase, not drug, that compound from a mushroom that specifically inhibited RNA polymerase 2. Here's a picture. I've never seen this picture on the exam, so I'm going to move past it. Transcription factors, I already talked about that. There's more details here, but I gave you the general information that you needed. They are there to help regulate transcription. You need them in order to recruit RNA polymerase 2, or technically any RNA polymerase, to a particular site in the genome. Okay, so some biology stuff. Let me point out some cool things here. All right. Probably more important for the MCAT than for this class, and maybe if you're taking a cancer course in the future. This slide describes a set of genes called tumor suppressor genes. These are special genes whose main job is that if they are working, they prevent the development of cancer. That's why they're called tumor suppressor genes. A lot of them are transcription factors that regulate the cell cycle. What does this mean? Well, a good example is P53. P53 is a transcription factor whose normal job is to prevent the cell cycle, is to prevent the progression in the cell cycle unless everything has been checked. It's kind of like the master regulator of cell replication. Basically, remember in Bio 1 you learned that there's like the different phases in a cell cycle, G1, S, G2, all those things, and then mitosis, right? There are checkpoints in between each of those stages that to make sure that there isn't any mutations in DNA, that DNA is being sorted correctly and replicated correctly. P53 is a particular transcription factor and tumor suppressor whose job is to make sure that the cell cycle does not continue forward unless everything is good, unless there's very few mutations or anything. This is why, very often, if we are studying cancer and we take a tissue sample from a cancer patient and we look at their genome, we find that p53 happens to be mutated in that cancer sample. That makes sense. If p53 isn't working, it can't make sure that everything is going right during the cell cycle, and therefore you get mutations and a bunch of bad things. So there's a bunch of genes here that are usually found mutated in cancer patients. Like this one here, my professor used to study, called RB1. If this one is mutated, it leads to cancer of the eye, retinoblastoma. There's a bunch of examples here. You might recognize these, BRCA1 and BRCA2. They're similar. If these are mutated, they lead to breast cancer. Overall, everything listed here is a tumor suppressor gene, again, whose job is to, if they're working properly, prevent the development of cancer by inhibiting the cell's ability to replicate and proliferate. Let me see anything else. You can read extra details there, but those are the basics. Let's see. Oh, yeah. All right. More compare and contrasting with prokaryotes. In prokaryotes, transcription and translation both occur in the cytoplasm, right? That gives it some advantages and some disadvantages. Because both transcription and translation occur in the cytoplasm, when you transcribe an mRNA, it can begin to be translated immediately. In eukaryotes, we have a problem. Transcription occurs in the nucleus, but where are all the ribosomes in eukaryotes? They're out in the cytoplasm. So in eukaryotes, we have to transcribe an mRNA and ship it out of the nucleus. We have to do a bunch of modifications to that mRNA in eukaryotes in order to keep it stable so that it can be shipped out of the nucleus, reach a distant ribosome, and be translated into a protein. Some modifications we see to mRNA in eukaryotes is attaching a poly-A tail. It's literally a string of A's at the end of an mRNA sequence in eukaryotes. So in eukaryotes, we learned way back in like 10b that, you remember that? Actually, what mechanism happens in eukaryotic transcription that we don't see in prokaryotes that you learned way back in chapter 10b? Before the RNA editing stuff, it starts with the S.
Splicing.
Splicing, right? Splicing is a feature unique to eukaryotic transcription. How does that work? Well, in eukaryotic transcription, we create what's called pre-mRNA, and then in order for transcription actually to finish, we splice out some stuff from that mRNA. I know it's a long time ago. Does anyone remember what we splice out in eukaryotic mRNA? We splice out introns, and we keep the exons, because the exons are the ones that actually code for important things, that actually code for proteins. So when we splice out the introns and we put together the exons, those are some things unique to eukaryotic transcription. Here's another thing that I might have only briefly talked about that many SI sessions ago, is that we attach a poly-A tail to it to keep it stable. There's usually also one more thing they talk about. It might not be on this slide, but we also usually attach what's called a 5' G cap, or guanine or guanosine cap, to these mRNA strands. That cap has several different functions, one of which is to provide stability. Another one, and it'll be detailed somewhere later, is that it also provides kind of like a signal for ribosomes to bind to eukaryotic mRNA. When we get to that, I'll point it out. But those are all these extra things that happen in eukaryotic transcription that don't happen in prokaryotic transcription. Some unique application to this, by the way, a nice biomedical biology application to this, is that if every single mRNA has a poly-A tail attached to it, in biology, we can design, I don't remember exactly what they're called, but we can design these hooks that are basically just a bunch of T's put together. And what we can do is in this sample of mRNA, we can extract eukaryotic mRNA out of this sample by just creating this hook of a bunch of T's, and they'll grab onto these poly-A tails, and we can just pull them out of solution, out of this sample, and do experiments with them. So that's something, again, unique only to eukaryotes. You don't see that in prokaryotes, and that's because of this poly-A tail. Let's see. No. Let's see. Make sure. Whoops. Transcription. Yeah, no. This is new to me. I will go over it during the exam review if it's necessary, but I am going to skip it now because there's plenty of cool things on here that I do not have time to cover. So, HIV stuff. Sometimes he gives you questions on HIV and how HIV is so hard to treat. Those, unfortunately, are in slides that are scattered out throughout your notes. I think there was some HIV information at the end of 11. There's some at the end of 10B and everything. And that'll give you some information on why HIV is so hard to treat. You're going to have to go back and look at each individual thing because I never remember all of them. I only remember a couple things. One, HIV is so hard to treat because it mutates so often. Why does it mutate so often? Does anyone remember what type of virus HIV is? Is it a DNA virus or RNA virus? It is an RNA virus. There's a special enzyme that all RNA viruses have in order for them to insert their genetic code into a host cell. What enzyme is responsible for that? Reverse transcriptase. Okay, keep that in mind. Any virus, and any RNA virus, and especially HIV, because they have reverse transcriptase, they are able to take their RNA genome and insert it into a target host. That reverse transcriptase is not great. It makes mistakes all the time. And now, usually, for many organisms, if an enzyme makes a mistake, that's bad. But for HIV, it's a survival mechanism. It's an unintentional survival mechanism. The fact that reverse transcriptase makes mistakes as it's reverse transcribing an RNA genome into DNA gives, every time HIV uses it, creates a slightly different genome in the host cell, which is bad because if it's mutating that often, we can't design drugs that specifically target maybe the enzymes HIV makes, maybe the proteins that HIV makes. It's very hard to actually design anything for something that mutates so quickly. That one is the only one I know off the top of my head with why HIV survives so many different things. You'll have to go through the notes to find some other stuff I usually can't really remember. Oh, I guess this is one here. HIV uses an RNA genome to have a cap and a tail, enabling them to mimic harmless messenger RNA of the host. So, if you don't know what they mean by cap and a tail, we just went over that. In eukaryotic mRNA, we have some extra things we attach to it in eukaryotic transcription. That is attaching a poly A tail and a 5' G cap. I guess I never remember this fact, that HIV has that feature as well, making it harder for the cell's own mechanisms to identify it as foreign genetic material. Okay, and that's the end of 29A. So, question, yes?
So, in terms of those transcription factors, we don't
Yes.
need to know the specific one and what they...
Yeah, which one binds to what? Yeah, no, the only one that sometimes is asked about is, let me pull it up for you, is TF2D. Yeah, this one right here. Let me zoom in. Is this one, transcription factor 2D. And that's because it likes to bind to the TATA box. Most genes contain a TATA box in their promoter, not 100% of them, but most of them contain a TATA box in their promoter. Yeah, there's other details on here. Historically speaking, I can say that rarely they sometimes ask, which transcription factor likes to bind to the TATA box? Answer is clearly here, transcription factor 2D. All right, let me see. I'll go over elements in 29B in a little bit. All right, so let me open up 29B. Okay. They start again by reiterating the mechanism of transcription in eukaryotes. Oh yeah, there's a couple extra things. Don't remember yet because this is more stressed in chapter 30. But in prokaryotic transcription, there's a particular sequence that's on mRNA. After transcription is done in prokaryotes, there's a sequence called a Shine-Delgarno sequence in prokaryotes. It is emphasized more in chapter 30, but basically the function of that sequence is to be a landing site for ribosomes, right? So after you're done with transcription, the next mechanism you have to worry about is translation. So the way that's sort of regulated in prokaryotes is that there's a special sequence on prokaryotic mRNA, called the Shine-Delgarno sequence. That sequence's job is to be like a signal, a landing site for ribosomes to bind to and to translate. The reason why I bring that up is because in eukaryotes we have something similar called a Kozak sequence. Okay? It's called a Kozak sequence. It is a region in eukaryotic mRNA that, oversimplifying it, is involved in ribosomal binding in eukaryotic mRNA. Okay. And then the rest of this diagram we've already went over, so let me move on to the next thing. Oop. Okay. All right, let's see. This technically also exists in prokaryotes, but they go over it more in detail in eukaryotes, is that in transcription you create, oh yes, question.
Real quick, so the cosexions and the shine-delgarno,
Mhm.
those are where they start translation?
Not where they start, but where ribosomes bind to. And then where they start translation is usually right after those things. But again, I know they bring it up here. That's more talked about in chapter 30. But they use the names here, so I have to give you a short explanation of what it is. But yeah, the main function of both of those sequences is it's a place for ribosomes to bind to the mRNA so it knows where to begin translating the mRNA. Okay, so on that note, this kind of implies that the entire mRNA will not be translated into proteins, that only part of it will. You will learn in chapter 30 that usually, well, all the time, that translation begins at a position on mRNA called a start codon. That is not usually the first base in an mRNA. That's usually somewhere downstream on the mRNA strand that was made. This means that there's a region on mRNA called the 5' untranslated region. This is a region on mRNA that is before the stop codon, and I know that's a chapter 30 term, but they're basically saying that not everything is translated in mRNA, that there is a particular region on it that will not be translated, and that is called the 5' untranslated region. This is where you would find things such as the Kozak sequence and other really complicated regulatory elements that regulate how mRNA is metabolized in eukaryotes, but don't worry about that. Bottom line is there's a region on mRNA that isn't translated sometimes. That is where the Kozak sequence is. If you see anywhere, that it won't be asked about in this class, but from here and in the future, if you see the phrase ORF, that is what is called an open reading frame. That's a complicated way of just saying the space between a start codon and a stop codon. So it's like a different way of saying the region of mRNA that will be translated. Okay, if you see the phrase open reading frame. Let's see. Okay, so now we go into the specific, like, special terms they use when they talk about eukaryotic transcription. One of which is they use the phrase elements. Elements are those consensus sequence things we've been talking about. They're the umbrella term to describe all those consensus sequences that transcription factors will bind to. That means they include the sequences in promoters, both the core promoter and the regulatory promoter, and sequences that you find in enhancers, okay? We'll go over these extra terms in a bit, but basically you see this phrase in blue, elements. Whenever you see the phrase DNA elements, they are DNA sequences that regulatory proteins like to bind to, meaning that transcription factors like to bind to. So they like to bind to promoters, enhancers, and these other unique terms called response elements, silencers, and initiators. I believe there's more detail given on them. If not, I'll give you a very short explanation of what each one is right now. A response element is still a DNA sequence that transcription factors like to bind to. However, transcription factors only bind to response elements in certain environmental conditions. The one example I gave was a low oxygen environment. So You can have transcription factors that will only bind to response elements in a certain environmental condition. Silencers are specific sequences that when a transcription factor binds to it, it actually inhibits transcription of a gene. So I may not have told you guys that, but transcription factors don't always activate transcription. There's a small subset that in certain conditions will actually turn off transcription. Those are transcription factors that bind to silencer sequences. So you can guess that the phrase initiator sequence probably means just any general sequence where when a transcription factor binds to it, it promotes transcription. Okay, so there's kind of like synonyms and overlapping terms that you can use. You can say that there's a promoter sequence there's a promoter region that has an initiator sequence in it. You can say that there's an enhancer region that has response elements in it. There's a lot of overlap here. Yes, question?
Do we need to know that slide with the initiators from a couple chapters ago, like ORC and MCM and...
Oh, for each of those genes? No, if we're thinking about the same thing.
Well, you said it,
mhm.
you were used to that slide having something that says skip, but that it didn't have skip.
Was that in prokaryotic transcription? I can flip through it real quick.
No, it was
Or eukaryotic replication.
eukaryotic replication.
Yeah, that one. I know which one you're talking about. I can't describe it, but yes, that one is still a skippable slide. The only thing you need to know for eukaryotic replication were the enzymes, the polymerases, and the thing with telomerase. Those are the only things you need to know with eukaryotic replication. Let's see. Okay. Other cool things, the eukaryotic transcriptional regulation. We already talked about this, the general transcription factors, right? Those are the ones that bind to sequences in the promoter region. And then remember that eukaryotic genomes have what are called nucleosomes. Does anyone remember what that is? That's DNA wrapped around what? Histone proteins, right? That's only a eukaryote thing. And what that means is you have regions of the genome that have a lot of nucleosome density and other regions of the genome that are very low in nucleosome density. This creates patterns of gene expression where some genes are expressed really highly because there isn't a lot of nucleosomes getting in the way, and some genes that aren't expressed very often because nucleosomes are very dense and preventing transcription factors from getting in. It turns out there's a class of enzymes, this is one of them, called histone acetyl transferases. You can guess by the name what it does. This one will attach an acetyl group to a histone protein. A long time ago I said that acetyl groups do what to gene expression? Increase or decrease it? They increase. Acetyl groups increase transcription. So there are a category of enzymes whose job is to go around and attach acetyl groups to histones, acetyl groups to nucleosomes to increase transcription. You can bet, it's not listed here, that there are methyl transferases whose job is to go around attaching methyl groups to everything, thus inhibiting transcription of whatever genes it happens to methylate. All right? Make sure, yeah, it's still going. Okay. Enhancers, we talked about it. These are those regulatory DNA sequences that are really far away. They use the phrase distal to describe that. Similar to promoters, both the core and the regulatory, transcription factors bind to these things as well. Some cool things about enhancers, though, is that they can be both upstream or downstream of the gene. Contrast that with promoters. Promoters have to be upstream of the gene. Okay, by definition, promoters have to be upstream of the gene. Enhancers, because of that unique feature of being so far away, they can be upstream or downstream. And crazily enough, a single enhancer might work with several different genes. One enhancer might be regulating several different genes simultaneously, depending on what transcription factor binds to it or what genes happen to be nearby. And yeah, it even states here, it depends on what specific transcription factors bind to that enhancer region. So you see there's a lot of crazy stuff with enhancers, why a lot of research is being put into studying them. I talked about silencers already. Those are the sequences that when transcription factors bind to them, they inhibit transcription. And response elements, same thing. DNA sequences that specific transcription factors, when they bind to them, are in a response to a particular environmental condition. Another example given in the notes here are heat shock response elements. These are ones that respond to sudden changes in temperature. All right, and there's a couple other ones listed here. We'll have to end for now. I will finish this up next SI session, and I will see you guys then. I'm going to try to have the review sometime, I don't know yet, Tuesday, Wednesday, or Thursday next week.